This gives you all my information. So if you want to get hold of me in any way, please feel free. Happy to take questions and happy to answer questions. I can't answer all of them, who knows, but at least I might know somebody who can answer them. That's always the way it goes. We are going to talk about, this was rooting, so we're going to talk about roots. I'm glad that I guessed, when I was driving up here, I didn't realize how many orchards and vineyards there were. So I threw some things in to talk about the importance of some of the things I'm talking about in orchards and vineyards. The one thing that I'm going to say right from the beginning is that the principles of soil health are all the same. It doesn't matter if you're farming 1,000 acres or if you're in a backyard garden, it's your agro-ecosystem, and it's all the same. The foundations of soil health are all the same. It's about living soils. It's about creating living, healthy soils. And the way it works, if you're working with trees, well, then there are some other things to consider. If you're working with field crops, there's other things to consider. But diversity is still going to be one of the foundations, and having roots is going to be is your biggest foundation. And I'm going to turn this around so I can see it. I thought I'd throw this in because, for one thing, I always ask people, and I'm not going to get a chance to ask everybody here right now, but what is Regen Ag? For me, it's just kind of a buzzword. And Gail Fuller, who is from Kansas, from Severy, Kansas, I think Gail nailed it when he said it's life. I think, everybody's regenerating something, but this is sort of the 6 core principles, which you'll also recognize from conservation agriculture. They are exactly the same. It's all been borrowed and sort of cobbled together, woven together, if you want to call it that. And I think in the end, you'll see that the underlying all of these things is just soil health. We have soil health, good healthy soils, biologically active soils. We have it all. So I put the context down because I think the only thing that nobody understood, including me, was what's the context of all of this? And I think putting it into context was important. But I think the thing is, you should be able to answer this in 3 to 5 minutes if anybody ever asks you. What's your mission? What's your vision? What's the goal for your farm? What's the goal for your agri-ecosystem, even if it's your backyard? What is it? What are you trying to do? But today, we're going to focus on this. I put biology at the top of soil health because biology is what really unites the chemical properties of soil with the physical properties of soil. And you'll see that as we go through. And then we talk about soil productivity. Well, if you're in large-scale agriculture, you're going to measure that in yield. You're going to talk about bushels, how much yield did I get? But that would be the same with anything that you're doing. It would be how many grapes, how many tons of grapes did I produce? How many tons of apples did I produce? But I'd like you to think about it a little differently. I'd like you to think about the fact that you have an agro-ecosystem. So is my ecosystem leaking? Am I losing chemicals? Am I losing nutrients into the environment? Because if I am, that's a bit of a problem because I want to hold them all where I am. I want them all to go into my food quality. And that's something that I've been working on. For the last 4 years, I've been living in Boston. And I'm going to move from Boston in May. I co-founded a company called Audacious and built a very fancy analytical lab to analyze food, to look at food quality. I was hoping that my vision for the company was that we would be linking how we grow food to the nutrient density in food. Didn't quite go that way. And also I got tired of being tethered to a laboratory. I needed to get my hands dirty and I needed to be back on the land. I mean, it's just, I said in my LinkedIn post, I needed to make my heart sing and feed my soul. And being in a laboratory just all the time didn't do that, as you can probably imagine. So, yeah, it can just go away. I think it will go away, hopefully. Let me bring this down here and just go cancel. All right. And then ultimately, if we can reduce the amount of nutrients that are leaking from our system, we're going to have health for ourselves and our animals. Now, if you're doing a lot of tillage, now the gardens are a little different, and I'll sort of couch this a little bit. If you have loads of organic matter, your system will be a little bit more resilient, and you can do a little bit more tillage and get away with it without doing the damage that you would be doing if you didn't have a lot of organic matter. But ultimately, we want to stay, and I work a lot in Amish country in Indiana, and I really don't care how you do low disturbance. It's about low disturbance. When in Lesotho, everybody's doing they've got little hand no-till planters. But the whole idea is to keep things covered, keep roots in the ground, and let's just break there for just a minute. What is the most available source of organic matter in your soil? It's not the residues. Residues sit on top, right? And then they break down, and then some of the carbon goes in. It's your roots. The roots are already there. So it's about decomposition of the roots. It's about decomposition of the microbial biomass. And in fact, what we know is that, and the biggest equation part of sequestering soil is microbial biomass carbon. So it's about roots and how many roots I grow and the diversity of compounds that are in those roots that are going to grow my soil carbon. And the best way to do that is to be doing these kinds of practices where I'm minimally disturbing the soil. And we're going to see why as we go forward. Healthy soil is a habitat. And that's the key part about all of this. You've got healthy soil. If you've got healthy soil, then it's a habitat. Things are living in there. Things are growing in there. They're building infrastructure. And if I'm going to define the easiest definition, easiest characteristic of a healthy soil is structure. You should have good structure. If you have good soil structure, it's not blowing away, and I'm not going to talk about soil wind erosion, because I know everybody here has seen a lot of that. We didn't get that down because most of it's rangeland, so we didn't get sort of the windstorms. But I've seen them in Spokane and whatnot, and it's terrible. And it's just, really, we have to think that there's no excuse for losing soil. I mean, we have too much technology to lose soil. The other thing is when we have healthy soil, our soil looks like that. It's got infrastructure. So we go and till all this stuff. What are we doing? We're wrecking the infrastructure. And who likes road construction, right? Such an inconvenience. And think of all the energy and all the costs to the taxpayers and everything to rebuild roads and do that. Well, think of all the energy all these little organisms are using to rebuild their infrastructure when we destroy their habitat. So try and think sometimes in terms of, what you know. So let's look at this. Rooting. Plants don't have to use so much energy to root in good soil. They don't have to push through. They don't have to barge their way open. They don't have to use energy where they can only grow a couple of roots because they're pushing down. Instead, they can grow a lot more roots. So what happens when they grow a lot more roots? They have access to a lot more nutrients. They can build soil structure a whole lot better. So now all of a sudden, everything starts cascading in a very, very positive way. And that's what we're looking for. And we start to have really good soil structure, and we start to have healthier plants that grow faster. And that's what we're really looking for, because everybody with climate change, we're all struggling. It's either too hot or it's too cold when we start. So we want to have good plant establishment. Well, if we want good plant establishment, we have to have good soil structure to do that. Now, the really good news is about having highly organic soils, or just even if you can add 0.5%, you're going to add degrees of temperature. Because the more active your soil is, the more it will warm up. I mean, there are people in Canada that, with really cold temperatures, and in North Dakota, that are seeing the advantage of having active soils, where their soils actually warm up earlier than everyone else's do because of the activity in the soil. And you can have runoff on rangeland if it's not grazed properly. And with small holdings, this is so true. When I was living and working in Montana, I used to ride endurance. And a lot of the women who had small 20-acre parcels had too many horses and just grazed it and living by rivers. And they're going, oh, I don't have erosion. I would do this. And then they're like, oh my gosh, what do I do? And we started rotational grazing the horses. And it was amazing what we could do. All of a sudden, the horses, electric fence, and the horses were moving, and they were moving all the time. And we were seeding and irrigating behind them. And all of a sudden, there was plenty of forage, and we didn't have to buy hay as much. So there's lots of things you can do. This is one of the easiest ways to measure soil structure. Just recycle drink bottles. It's not that hard. And all you're doing is taking a core of soil, putting it in the top, running water through, run the same amount of water through both sides, and then look at the soil structure. This one has completely collapsed, and I've run it all off. This one's nice and clear with a little sediment. I know that this one has good soil structure. So now I can tell that I don't need somebody to go and tell me what my soil structure is. I've measured it myself. I know that I'm doing okay. I don't need to use fancy rainfall simulators or anything like that. This is just an example of multi-species versus single species, because people ask me, said, I covered everything in rye, or I just did one. 1 is better than none. 2 is better than 1. 3 is better than 2. And so if we can just get 3 or 4, yay. But let's have a look. I think you can see the difference, single and multi. And part of the reason that is that every different plant species you put in the ground leaks its own unique signature of carbon. And that signature, everybody thinks sugars right away. Sugar, sugar, sugar. It's not sugar. It's amino acids. It's organic acids. It's lipids. It's complex compounds. It's terpenes. It's polyphenols. It's lots of things that have big, complicated carbon chains. And the more complicated they are, the longer they stay in the soil, the slower they take to decompose, and the more carbon you're building, the more food you're building. And the important thing here is that these organisms want nitrogen. They want trace elements. They don't want sugar all the time. They need a bit of sugar just like we do. So they need some carbohydrates, but mostly they need protein and amino acids. And they're going to choose amino acids over protein, because with protein you have to actually spend energy to break it down, right? But with amino acids, I can just combine them right into my body right away and get the benefit of them. So think of your soil as a habitat, and you are stewarding it. And I like to talk a lot about agri-ecosystems, because I think that's what we're all doing. We're all stewarding an agri-ecosystem. Whether we, enjoy the concept of an ecosystem, think it's too green focused or whatever, but you have an agri-ecosystem. So let's just think of it that way. And your soil must be alive. This is a handful of no-till soil in Lethbridge, Alberta, Canada, growing in 4 to 6 inches of rainfall. So, everybody thinks they know dry. There's all different shades of dry. And what's drought to somebody is not drought to someone else. But you can see, even in that system, just no-till and a simple system, and you can actually build soil. This is bacteria, and this is a really simple thing to do. All I did was just take a teaspoon of soil. I added water. In fact, you can do it off if you're doing your soil structure. I just took a little drop, and I put a little bit on a slide. I did a gram stain so that I could see gram positive and gram negative. And that was what just was in the water. So you can imagine what, if I could look at all of it, what it would be like. It's fantastic. This is Bacillus thuringiensis. This is BT. This is, and those crystals that are in here, these ones here, the little shiny bits, those are what they actually use the genetic code for into BT corn. Now, every healthy plant is trying to attract Bacillus thuringiensis and various other Bacillus species. Now, the reason they want bacilli is because they're highly antiseptic. They kill lots of other things, so yeah, they want them. So it wasn't ever a problem with plants attracting them, but now we put it into plants. The problem now is that because it's there, the plants don't really need to send out those root exudates and those signals anymore to attract them. And in fact, those plants don't do that. Fungi, a lot of people are thinking about diseases, and we all do. Fungal diseases, prevalent, cause lots of problems. But 85% of the fungi in the soil are just there to decompose things. The woody corn stalks would never break down if it weren't for fungi. Trees in the forest would never break down without fungi. We need the fungi to break it down, but we also need fungi to hold the soil together, to give it good structure. All those fine silk threads in there, what are they doing? They're making nets, and they're holding the soil together, and it just keeps going and going. We're getting bigger and bigger and bigger aggregates, and the soil is starting to really have good structure. We need to have fungi in the soil. Now, we're going to talk in a few minutes about mycorrhizal fungi too, something that's really beneficial as well. Actinomyces. People have seen this, and I've had a number of people say they thought this was mycorrhiza. It's not. This is actinorhizal fungi. They could be fungi, they can be bacteria, they're a combination of both. Every now and again, they reclassify them into something else. They're actinomyces is what they are. And they're also highly antiseptic. Now, there are also streptomyces that cause you problems on potatoes, which is why you need the good kind and not the bad kind. And the good kind always compete against the bad kind. And all your pathogens are really lousy competitors. So the thing is if we create good habitat, our plants grow really quickly, and we allow the plants to do what they need to do, we will have soils that actually protect plants for the most part. This is mycorrhizal fungi on a root, and you can see the spores here forming, and then all the hyphae around. Now, mycorrhizal fungi actually leak a substance called glomalin, and in leaking glomalin, they actually glue soil particles together. So not only does their hyphae forming these nets, but now they're also gluing the soil particles together. So now we really start to have really good soil structure. So now we're starting to really build soil structure. This is what they look like inside the plant. So if you're looking at mycorrhizal fungi, you really do have to stain the roots. I'm going to show you another way to look at it, but you have to stain the roots to see inside. And these are the structures called arbuscules where the exchange happens. And when mycorrhizal fungi colonize a root, they change everything about the plant. So now they change the photosynthetic capacity. The plant revs up photosynthesis. It provides more zinc, more trace minerals into the plant. It confers drought resistance. It confers disease resistance. The whole rhizosphere changes to be plant growth regulating. So it's all about promoting plant growth. And it's totally selfish, right? Because it needs a host to grow. So if the host dies, it's in trouble. So it's going to do everything that it can to make that host the best that it can be. And so we want these mycorrhiza fungi to be part of our plant system. And they will be. We don't have to add them. Not necessary. I mean, their spores will rest in the soil for hundreds of years, actually. And they will regrow from bits of plant roots. So the thing about it is when we are not doing a whole lot of disturbance of our soils, they're always there, and they can always colonize the next plant. Protozoa and nematodes. What makes nutrient cycling in biological systems work is predator-prey interactions. And we don't talk about that enough. Everybody talks about the microbes. I need microbes, I need microbes, I need microbes. No, we actually need predator-prey interactions. We need protozoa, amoeba, nematodes to eat up the bacteria and the fungi, to turn them over so that the nutrients become available to the plants, and so that we can create more habitat. And the beauty of these animals is that they actually concentrate the nutrients in and around the roots as they're gobbling up the bacteria and the fungi. Now, we want them to gobble up bacteria because bacteria can grow a whole new generation in 18 to 24 hours. A plant can't even, has no concept of growing that fast. So they can actually sequester and hold on to nitrogen, sulfur, phosphorus, all the trace elements, no problem. Plants can starve. So you can have these great biologically active soils, and your plants are starving, and you're like, I don't understand. It's because we have to get predator-prey interactions, and sometimes that's because we've used a lot of nematicides. Maybe we haven't grown too many brassicas. So the one thing is that brassicas are like Red Bull to soil fauna. They love them. There's something about what's in them that just stimulates the growth and reproduction of soil animals. I can't tell you what it is. We just see it over and over again. Nematodes, people are thinking about plant parasitic nematodes. But you know, the amazing thing is that most nematodes aren't plant pathogens. In fact, if we're having root problems, if we're having larvae that are eating roots, most plants send out signals, really complicated polyphenols that actually are signals, like signals in the soil to bring in nematodes that feed on insect larvae and insect eggs. So plants are trying to take care of themselves, and the mycorrhizae are trying to take care of the plants. So all this is happening without us really doing that much. Collembola, or springtails, we see these, in the leaves, and you look down at the leaves, and the leaves are just lacy. They have nothing left in them. Collembola have had their feed. They just eat everything out like that. Now, they're part of the whole decomposition cycle. So what they're doing is they're eating holes in things, and then the fungi are going in and breaking down the really woody tissue. And so they're working symbiotically. And in fact, Collembola prefer to eat leaf material that's colonized by fungi, and they don't care if it's pathogenic. So from an orchard and a vineyard and any kind of tree production, fruit production, these guys, and having good soil detritivores is really important because they're eating up all the things that can cause us problems. So we want the earthworms to be burying our leaves. We want these guys to be eating up the fungi on our leaves. We want this all to be going together. These are microarthropods. This is how small they are. And you saw the one at the beginning. That's how big they are when they're magnified. Very large. But this is an enchytraeid. Enchytraeids are related to earthworms. They're very tiny earthworms. And once again, they're concentrating nutrients in and around the roots. They're not potworms, which are nematodes. They're actually earthworms. And if you look at them, even with just a small magnifying glass, you'll see that they're segmented. These are your earthworms. There's a cocoon. They can sometimes look like popcorn, small popcorn seeds. This is how they're made. So earthworms are hermaphroditic, which means they're both male and female together. The big night crawlers actually have to outcross. The ones, the earth workers don't have to outcross, so they can just self if they want to. But earth workers are the ones we really care about because they're the ones that are really mining our soil, really changing the structure, really improving the structure, and bearing lots of leaves. And in fact, the night crawlers, although they make people's lawns pretty hard, the night crawlers actually are doing the same thing. When they pull all the leaves together in a midden, that's a big compost pile. They're mixing their manure with the leaves, and the whole thing is decomposing. And if you actually could break that down, it's a whole little decomposing system all to its own, all unto itself. And then they rest. And so when the weather is too hot and too dry, they rest. So it doesn't matter if you're irrigating all the time and you're wondering, well, I guess I don't have any earthworms, but it's hot. They don't like hot soils. So they move down into the cold. Now there's a reason for that too, is because cold temperatures, more oxygen. Cold water, more oxygen. So they like colder temperatures. When it's too dry, they move down as well. And they round themselves into a ball like that. And they actually make like a hard case around themselves that is waterproof and airtight. So they can't decompose. And fall tillage absolutely destroys these things and cracks them open. And then we lose a lot of earthworms with fall tillage. So I tell people, if you want to do tillage, do it in the spring. Don't do it in the fall. You're not getting ahead of anything. Rhizosphere. The rhizosphere is just the root, the soil attached to the root, and the soil influenced by the root. And I already talked a little bit about soil root exudate, so we're going to get into that just a little bit more. So this is the model of the rhizosphere I like, and the rhizosphere is really drives everything. It's the most biologically active part of your soil, by far. There is nothing that's more biologically active than the area where all your roots are. So if it's a row, it's active. Between the rows, not so much, unless you're growing companion crops or something. But in the row is really the active part. A prairie, the whole thing is active. And it begins with a seed. So every different seed you put in the ground leaks its own unique signature. It's trying to attract the things that it needs in order to grow. It's also trying to repel other things so the seeds don't get eaten. So there's a whole lot going on in seeds. Good seed is the most important thing you can do. Buying good seed, having good seed. Because your seed should contain everything that plant needs to grow in water alone for 10 days. And you can test it. Just put it into paper towels and water and see if it will grow for 10 days. It should grow for 10 days. If it doesn't, if it gets all moldy and everything else over that time, you need to work on that. And that's where you really need to do some seed treating, or you need to do some compost tea or something like that. Good seed has a beneficial spermosphere, and that's the area around the seed. This is where it all starts. As soon as that seed sucks up all that water, starts leaking things out, that's where everything begins. And it's either going to be positive or it's going to have some real disease problems. And we want, this is the start of it right here. This is where the exudates start. This is where the seed becomes really, really vulnerable, right? Because now you have the radicle, the embryonic root, and you've cracked open the cotyledons. Everything is really, really, so that's why the plant needs to have all these beneficial things going on around it so that it can't get diseased at this point in time. Exudates. The plateau for the exudates for emergence is as soon as this comes out. So as the seed's hydrating, this is when everything starts going. So seed is the best part of this. I love this shot from Jimmy Emmons. See these are all root hairs, crazy root hairs growing everywhere. This is what it should look like. This is a plant that's growing in healthy soil. It can afford to grow all those root hairs. It doesn't have an energy limitation. And think of how that's binding all the soil together. I saw this great picture. I was sent this great photo the other day of soil from the Kalahari Desert. And this farmer had been growing, sparse crops, trying to grow some forage for his animals, pulled up this plant, and the sand was just stuck to all this huge root system because of the root exudates. That's banana roots there. And you can see what we choose, different root architecture. At the German National Field Day every second year in Europe, they have all the seed companies have every different variety of every crop that they breed in the soil, and they have pits behind them where you can walk through and see the rooting. And you can see that the different varieties have different root structures. They have different depths of structure, different depths of roots, and things like that. So, and then you see the above ground as well. So you can actually really think about, well, what do I want? What is my goal? Do I want a few more roots and I can still maintain yield, or do I want this, do I want that? These are the things we need to ask our own breeders. And we want to see things like this. And these are really easy, these plates are really easy to do, but this is what the kind of thing is. Have a look at these roots. Have a look at the roots. Dig them up if you can. Persian clover, really drought tolerant. Look at the fine root structure. Woolly pod vetch doesn't have a fine root structure. So if I really need to build structure, maybe I don't want that one. I definitely want faba beans. Lots of roots. The plant will be like 2 inches high and just totally big, fat, juicy roots. Yeah, so if I'm trying, this is the things that you can use to build your soils. Use to build your nutrient cycle too, right? Oh my gosh, I'm just having, what the heck? Rhizosphere millet from Chris Nichols. You can see the mycorrhiza holding everything together. And then this is a picture of the mycorrhizosphere. If you don't have mycorrhiza, look what you're missing. Look how much soil structure I'm going to hold together. Am I going to have the dreadlocks that Christine Jones talks about? Yeah, probably. Sometimes you don't. Depends on the soil. Depends on the temperature. Depends on whether it's really wet or not, or whether it's too dry. Oh, I see what's happening. I am touching my pad the wrong way. This is what was happening in the Kalahari Desert with the sand. The mycorrhiza were all holding the sand particles together. We can easily build structure. And you can see the root here, and now you can see them holding everything together. And then this is just an example of the variety of spores that would be in your soil. And that really depends on the variety of crops you're growing, the cover crop, the forage crop, the companion crop. Weeds are very mycorrhizal. This is knapweed. Totally dependent on mycorrhiza. You can actually see mycorrhiza. They have, when a root is colonized by mycorrhizal fungi, it actually forms a yellow color, a bright yellow color that is actually photosensitive. So that color will disappear if it's out in the sun for too long. So if you take the root out and you kind of dip it in water really quickly and pull it out, you'll actually see the yellow parts of the root. And that will even persist later on in life. And you'll actually get to see the yellow color. And you can actually see I didn't get a complete control because it's actually colonizing the root there. Onion roots, if you are interested in doing your own assessment of how much mycorrhiza you have in your soil, baiting your soil with onion roots is the easiest way to do it. Onions are nice straight roots, and they come out of your ground. If you mix your soil with a bit of sand, like half and half, and then just grow it in there and then pull them out, rinse them off, you'll be easily able to assess the yellow color, and you can get a percentage. And you can say, oh, this one's 50%, this one's 20%. So you can work on it that way. And then you can always look at it in the microscope if you have one. But you don't need one. And you can see the mycorrhiza even will mine old plant tissue. Like these roots are really yellow, and they're actually mining old alfalfa roots. So it doesn't stop there. They'll mine out the old stuff. And I think I've pretty much already said this. They change everything. Microbial community. They really prefer amino acids, so you get a lot more amino acids being exuded. And it really increases the rate of photosynthesis on these plants. So the other thing in healthy soil, if you're making the transition to, less disturbance, you can't give up on fertility. I'm going to tell you that right now. Because when you start building that whole big population of soil microbes, they eat nitrogen, they eat phosphorus, they eat iron, they eat all these things that your plant needs. And they're growing fast. And your plant needs it too. So if you start pulling back on fertility too fast, your plants will starve, and you'll start to see this decline in yield, or they'll all get lethargic, and they'll all start to be looking like they stopped growing. And part of that's because you just pulled back too fast. You need to maintain it for about 2 to 3 years. Not at the same level, but you can't just drop out. And you have to really watch that. And so I like people to say, try some test strips. And I know that's really hard because most farmers are like, so did you do a test strip? Yep, 100 acres over there. And I'm like, okay, not bad. That's a big test strip. Just, just do 1 row. That would work. But always do a control. Now, when I say control, a lot of people think of controls as having nothing. No, it doesn't have to be nothing. It can be what we call a reference strip. So it's whatever you do all the time. So if this is your normal practice, then your new alternative practice should be beside it, and you just make the comparison between the two. These are the plants that are highly dependent on mycorrhiza. These ones not so dependent, but they grow them. And then your non-hosts are over there, and your brassicas do not host mycorrhiza. They have their own way of getting phosphorus and other things out of the soil by leaking a whole bunch of acids. And so, but the other thing to remember is that most of your vegetable crops are highly mycorrhizal. Potatoes, carrots, all of them. So be thinking about that when you're growing veggies. And apples are also mycorrhizal. And so are grapes. So let's dig into the rhizosphere effect and see what we're going to look at here. These are all the things that go on in the rooting zone. We can use all of these things to our advantage. All of them. So, people grow rye. Put a rye cover crop in. Why? Because you don't have weeds the next year. There's all these properties. Rye is this quick cleanup crop that everybody knew from a long time ago. You plant rye, you get rid of everything. It can also be in our dry climate here, at least in southwestern Kansas. I had a great rye cover it was multi-species. And it wasn't a year after, but it was 2 years after, in the second year, I planted milo into there. And every time it rained, the milo would stop growing. I'm like, what the heck? And then as it would dry up, the milo would grow like crazy. And I thought, what? That's not right. This is the reverse. With water, it's supposed to grow. When it's dry, it's supposed to, then it can stop. No, it wasn't. So I went into the field and I was digging around and I realized I still had all this rye residue in there. I mean, it was a lush crop. And every time it rained, all the allelopathy just leached out into the rooting zone. And the milo was like, oh, this is bad. And it would stop growing. And then as it would dry up, we wouldn't have the allelopathy anymore and grow like crazy. So just think, so again, we can use these things, but we have to think about how we're using them. We can use plants to control weeds. I can use various brassica crops to control nematodes. I can use my brassicas to also grow soil fauna. So I can actually start using plant species and manipulate a lot of things while I'm building soil health, while I'm maintaining soil health. And roots. So we think back to my model. Roots can actually change the soil structure themselves. If they need to hold soil next to them because things are getting a bit dry or whatever, they'll actually pull the soil, they'll tightly aggregate the soil. If it's wet, they can loosen up. And some plants generally have looser soil, so you grow a legume like peas in that soil will always be nice and loose and fluffy and whatnot. But they can actually control it as well. Again, a change in exudates, a change in the amount of exudates, changing microbial community. All these things cascade and are beneficial. Or they can be not so beneficial. But again, our healthy soils, foundation of agriculture, we have good soil structure, we're going to limit erosion, increase nutrient cycling. We're going to biodegrade toxic compounds because the more biologically active our soils are, the more they'll break down things that are in our soil. For example, glyphosate, glyphosate. If you don't use too much phosphorus, then the organisms in there will just break off the phosphorus and eat up the phosphorus out of the glyphosate. So it will break it down pretty quickly. So there are ways to be doing some, biological soils have a lot of advantages. But we also need to have some diversity. You can't always get cover crops in. I know in the Pacific Northwest we really struggled, in Canada we struggle. I really like companion cropping. So I'm growing plants between the rows of my crops. And I'm growing my diversity between my rows. And sometimes it's another crop if I'm intercropping, but if I'm not, then I'm growing as much diversity between my rows as I can. And that would be included in the garden. And the reason for that, again, if we think about it, is all the diversity of exudates. Now I have pretty uniform, but now I've got diversity. I've got different compounds going into the ground. I'm building my soil ecosystem services. I grow chickpeas with wheat quite often. These are Desi chickpeas, the more wild type chickpeas. They grow lower, they're highly mycorrhizal. Deer do like them, but I haven't had weed problems when I use them. Like people go, well, if you have a companion, what do you spray? It's like, well, you're not supposed to spray. That's the idea. The idea is that you don't spray. But, I'm also trying to feed my wheat. And people go, well, is it, I had some greening of the grain, a little bit of greening, but mostly it stayed below the canopy. If I'm worried that it's going to compete in the canopy, I will seed it after my wheat is germinated. I'll do the same thing for corn. I'll leave corn to the V4, V6, and then I'll seed my companions in after that. This is intercropping. This is chickpea and flax. This is the no-brainer one. If anybody wants a no-brainer intercropping system, this is it. I don't know if it would be the same here, but in parts of the north, it's the one that everybody can do. This is in Nebraska, actually southern Iowa, and this is Chris Teachout, and this is Chris's experiment that we did with growing intercrops in corn. And we also did the Three Sisters. And for those people that are gardening, the Three Sisters works really well. Corns, beans, and squash. And you wouldn't believe how all of them love each other. They just grow. I mean, it's a bit tangly to get the beans out, but orchards. I had an orchard in Spokane, Washington, on Mount Spokane. And actually, I love orcharding. And a lot of times I was just in California with grapes, and it's like, well, what do we put between the grapes? I like to have the whole row covered. And I think the same with almonds and everything else. It just prevents dust and erosion. And it's not competitive. It really is not. There's no evidence for that, as long as we do the right thing. Now, we also have to pay attention there. You can't grow a lot of clovers between fruit trees. Now, there's a reason for that, or nut trees. Root knot nematodes and clovers is this great host for root knot nematodes and other parasitic nematodes. So you start having problems with nematodes, you need to change over to a mustard or a mustard mix in order to back off on the nematodes. So I recommend that you always grow some brassicas, not a brassica total, but a brassica mix with your clovers and whatnot, so that you just can't build up nematodes. And if you can, you want to integrate sheep or something small in there to do that. It works really well. And we've been seeing that in the vineyards. We've been doing it in the orchards. In the fall, I would put pigs in my orchard because apple finished pork is to die for. So good. And there are pigs that come from England that actually, they like being in the orchards. And they don't root things up, and they just eat up all the culls. They manure your orchard, and you just have so much more decomposition. And I just didn't have the same fungal diseases that I would have had otherwise, or codling moth or things like that, because they were eating all the cores and eating all the old apples. Onions, and this is sweet alyssum. Everybody knows this. It grows in all the pots, and you go buy something. Sweet alyssum is an excellent pollinator, and it grows really low to the ground. Seed is cheap. It's really fine, so it's kind of hard to seed. But you mix it with rice hulls, and it does a fine job. And we grow it with onions because then we just get parasitic thrips, and that gets rid of our other thrips problem. So we're growing beneficial insects, and we're using sweet alyssum, and we'll use that a lot in vegetable production and low-growing flowers. This is carrot seed production. You can see the seed heads, and you'll see on the other outside rows are lentils. What we're doing is we're baiting, not baiting, but actually using the lentil rows to keep predatory insects there. And because ligas are really bad on carrot seed. This was organic carrot seed for bungi seed, and the ligas just suck the juice out of the seeds. And most carrot seed production is at like 60% viable seed, so it can be really low. When we started doing this, we were up at 80 and 85%, so it was really valuable. I think you could have done it with calendulas or any other flower in there just to keep other things away. I think the other thing that we know is that you don't have to have, if you have a solid one crop, okay, that crop row, when it's one solid color, let's say canola, or one canola field, solid color, when the flea beetles are flying over, they're like, yeah, I'm going there. If you just put like 16% barley in with that, nothing that's going to compromise the yield, it changes the color. And flea beetle damage goes down significantly. So it's worth considering. And once again, consider predator-prey interactions. That's what really drives all of this. It drives below-ground nutrient cycling. It does above-ground nutrient cycling and pest control. And when we have diversity above ground, we start to get diversity of more insects. We don't need to use insecticide, which can be incredibly damaging to a lot of things. And now all of a sudden, we have all these predators, and we don't have the same problems with aphids and thrips and things like that. I know there'd be vegetable production here, so mulching potato mounds. The potatoes that grew in the mulch didn't look very good at the beginning, but they yielded more at the end. So one message here is, this is a new way of doing things. Be patient. Watch over the season. Don't panic. You're like, oh, that doesn't look right, doesn't look right. I get it. Who wants to lose a crop? That's why I say one strip. If you're trying something new and you really don't know, just do a small piece, not going to cause you an economic loss, and watch it over the season. And so many times people watch it and say, oh, I wish I'd done the whole field. But you know what? You can do the whole field next time. But you never know. It's a crapshoot, right? It could have been really awful and could have failed, and then you'd be mad you did the whole field. We try and get ahead of it as researchers. We try and get ahead of it so that we can share information with all of you, so that when you try it, we've already tried it, we can go, yeah, don't do that, or yeah, you could try that, and maybe it'll work for you, maybe it didn't work for me. I can tell people that. Like, I'm the worst roller crimper person ever. Yeah, like, I don't know, I just can't do it. I grow these great crops, and then I roll them down, and they all stand up again, and I'm like, fine. And then I get the flail more out, and that's the end of that. You know, because we were doing that for squash and pumpkins, because it's great. But you really, I prefer to mow them. And now in the vineyards, we start doing the same thing. We have a thrower mower where we can throw both sides. And we mow and throw. We just, gave up the roller crimping thing. And I know some people it works great for. I just don't know. And I've been told, oh, you need more down pressure, you need more cutting bars, you need bars like this. Fine. I've tried them all. I suck at it. It's fine. Potatoes, and this is between the rows. These are companions in potatoes. And it actually influenced our potato yield. Influenced sugar beet yields too. So, you know, there's no end to this. When you have diversity, you have diversity of bugs too, as long as you're not using too much insecticide, or insecticide at all, actually. And then you start to get bugs that eat slugs. And this is the best one. These are these big crab beetles. And the first time I saw this was in France. And we'd gone to like a diverse cover crop mix, multi-species covers. And we'd done it for like 3 years in a row. And all of a sudden the slug problem disappeared. And we're like, wow, look at that. And then we started digging around, and then we saw these big beetles, and they're just chowing on slugs. It was great. And then you get birds in there too, and birds will eat slugs. And things like that. So think about the fact you start managing below ground habitat, then you start managing above ground habitat. And then of course, cattle. You know, why do you have to just grow silage corn? Who says that I can't grow everything in between the corn? Who said? The nutritionist. Well, yeah, because it doesn't quite fit the model. Well, that doesn't mean I'm not creating great forage. And this is the last part of Regen Ag, right? Integrating livestock. You put your livestock in, and people go, well, it doesn't pay. It pays. It pays up to $180 an acre. You can recover the cost of that forage crop fairly easily by growing livestock. And the benefit to your ground and everything is so powerful. But I understand that all people can't have livestock. I get that. So do a cover crop, do companions. Companions are your best choice if you're just a cropper. And this is the last part, and then I'm going to finish. I know there's a lot here, but I wanted to give you an overview. This soil microbiometer. So people always ask me, how do I measure? Soil microbiometer is a great little kit. Full disclosure, I'm part owner of this company. But I really like it. It's easy. You can do it on your tailgate. You can do it anywhere you want. Anybody can do it. It's not that expensive, and it gives you a really good idea of how much biological activity you have in your soil microbial biomass. So I recommend it. Solvita for soil respiration. Now you don't do it the way they say. You actually take the jar and put it in the soil where you're, like, actually do it in wherever you are. So you take the jar, you turn it upside down, you screw it in. And you leave it for a few minutes, because in disturbing that ground, you've off-gassed CO2 and nitrous oxides and things. Unscrew it, let the air vacate, put your tab in, and then screw it back down and record the temperature on the top and put the lid on. That is the soil respiration in your soil at the time that you're interested in it. Now, if you think about it, you send a test off for a test. They dry your soil, they grind your soil, and then they re-wet it. And 24 hours later, they give you the reading. Well, that's only the things that would grow fast enough. If you're actually doing your soil, you're getting everything. And I recommend you do it right beside the plants. Okay? You don't get plants in there, but you just do it right beside the plants. These tests here, Laqua sticks from Oriba. These aren't cheap, but they are amazing. So you pretty much do it like a sap test. So you squeeze the leaf. I like, if you have a lot of plant material in there, cell material in there, I like to put it through like a tea strainer. And then you put a drop in there, and these are all calibrated. They have a calibration solution with them. You can do hard water, you can do dissolved solid. I mean, there's a lot you can do with these sticks in the whole set. Phosphate, nitrates, the works. Now, these are what's called ion-specific probes. So it's the ions. So this is just like sap testing. It's the ions that are moving up and down. So you're actually measuring the ions. It's not total nitrogen or anything like that. It's the ions. Very powerful. They're not cheap, but they last for a really long time. And all the parts are interchangeable, so you just get new things when they actually wear out. I think that's important that our kids are our future, and nature deficit disorder is real. So anytime you can keep kids on the farm, you can bring kids from school on the farm, you can bring urban people out to understand where food comes from, I think it's really important. And we have a global food and farm community where we share information. It's, I say global because I really believe that just what I said this morning. It doesn't matter that I'm an orchardist. It doesn't matter if I have, 50,000 acres of grain. Keeping all those principles are the same. Soil health is going to be my friend no matter where I am. Thank you very much. And I think I still have time for questions. I'm pretty sure I do. Yeah, I got the thumbs up. Yeah.
So, great presentation. My question is, you hear a lot about soil health analysis, and had several occasions where I've talked to people at soil health workshops and their cover crops of work. They got a test for their nutrient application, but they had no idea what else is there. And I think they went and actually got on somebody's website and said, I'm going to get this cover crop and put these things together. So how do we encourage people to go past that first nutrient application test that you get from the conventional system and have them understand they need to know more than just what the next fertilizer is?
Oh boy, you're asking the $1 million question there. I think anytime there's change, I think part of it's the labs need to understand, okay? So I'm going to give you an anecdote with that. So one of my first clients was Jason Kim of Kim Potatoes, which is seed potatoes in Montana. And we grew cover crop, and then we were growing potatoes. And every time we put pediols in from that particular field, they would come back needing more nitrogen. They would. And we'd look, go, yeah, no. And then next week, pediols go in. Those plants are dying. Like, why are you sending me them? And we said, look, Montana State, we said, come out. Here's the map, go sample them yourself. Well, there's no yellow fields. Huh. Well, there has to be a yellow field. Those petioles are all, well, did they look yellow when they were in the bag? No, but they're deficient in nitrogen. They're not. Well, what did you do to that field? Well, it was a cover crop field. There is a difference in the way those plants grow. There is a difference in, and that's why, I mean, the respiration test is really important, but it needs to be done right. The other thing is that we talk about what's available. We have no idea what's available. The plant knows what's available because the plant will make whatever it needs available. So the best thing you can do is test your plants. And feed analysis is cheap. So do a feed analysis. You don't need to do a special tissue analysis, just do a feed analysis, depending on what you want to get out of it. But the other thing is that if you do totals in your soil, you get the totals from your soil, like total P and total that, you'll find out that you have huge amounts. Like, you'll see, I remember when I first really looked at soil tests, I was like, it says that I have 40 ppm of iron available. I'm on basalt soil. That's not possible. And so then I did total iron, and my total iron was over 100,000 ppm. And I'm like, well, I guess if the plant really wanted iron, it could get it. And then as a plant physiologist, I'm looking at this and going, I need to understand this more. And the point is that plants can actually get what they need. They will do, and I talk about this in another set of talks, but they can pump out hydrogen ions, they can pump out OH ions, they can pump out acids, they can pump out polyphenols, they can pump out any combination of them to get the ions they want. They will actually do really complicated molecules, polyphenols, which will chelate heavy metals, so that the metals are in an organic form that they can get later, so they don't leach. So the thing about it is that what we need to provide is the habitat. We need to make sure the trace elements are available in there, and then we need to just watch and let it grow. Sure, we need to tweak every now and again, but I think that, and I get it. I mean, my son went to university, and I need to make a living every year. I mean, I can't just, oh, it didn't grow this year. Oh, well, no income. No, I get that part. I really do. We can't afford not to have an income. And so that's the beauty of regen, though, is like if I'm an organic farmer and I work with a lot of organic farmers, I'm spraying organic chemicals because I can't afford to lose a crop either. But I'm still spraying, and many times I'm spraying way more expensive stuff to control things. And so once again, regen is this really nice, I think, hybrid system where we're taking the best of all these worlds and putting them together. And then just basically weaning ourselves off some of these things. We're going to have to augment fertility no matter what. I mean, you can't keep exporting grains and exporting your vegetables and everything and not expect to have to put some things back. But I think the labs, we really need, I think part of this is educating yourself and trusting yourself. Anna Kay, who's this woman from Oulu University in Finland, talks about farmer knowledge and the importance of supporting farmer-based knowledge and encouraging farmers to be confident in their knowledge. And I think that I don't know a farmer who can't walk into a field and go, yeah, there's something really wrong here, I can feel it. Okay, you feel it. There's nothing wrong with feeling it. Don't ignore that. You feel it, you feel that there's something wrong, start looking. Use your observation. Do that. Yeah, we're taught that you can't manage what you can't measure. I think that's rubbish. I think we can manage what we can't measure because we can't measure everything, and we can't measure everything perfectly or really well. And I think sometimes we're trying to measure at too fine a point. Do you really care that you're 1 ppm out? No, you should not. If you're 100 ppm off, you shouldn't care either. 200, 500, yeah, okay, I'm going to get worried. But 1 or 2 here and there, who cares? And that's the thing that we're not, you know, as a scientist I care. Okay, I've got fancy instruments and I care about parts per trillion. And if I'm doing heavy metals, I need to care about parts per trillion. If I'm caring about potassium and potassium's at 10,000 ppm, do I care that I'm 5 ppm off? No, I don't. And neither should you. So let's be realistic about how we look at these results and really think about it. You need them as an indicator, and always take them as an indicator. This is an indication. What do I see? I see everything going really well. Okay, well then, you know, maybe I look at this and I go, okay, this is pretty good, and I'm going to take this test and feel good about it. Yeah.
I've got some cereal rye growing on my raised beds in my garden.
Okay.
Should I worry about when the little happy comes down?
Yeah, you should. But no, it's your garden, so you can water and things like that. So, but you're growing that rye, and I mean, it's going to clean a lot of things up for you. I would make sure you kill it early and allow it to really break down. The other thing to think about, especially if you're having problems with diseases, especially fungal diseases, is think about instead growing like a forage oat. Because oats have these compounds in them that when they leach out, when the water leaches through the leaves, actually inhibit the germination of a lot of spores. So mix your rye with oats and then kill it early. Okay, well, you can do that too, but well, you want to leave the roots though.
Yeah, I know. The earthworms are there. They love the sweetness that the rye puts down.
Yeah.
No,
No, I didn't need to spray it. I meant like just watering and even cutting it, like just clipping it, mowing it, whatever you want to do. But leave the residues on there, protect your soil. Just give me a second with this one though, because with the orchards, I didn't mention this. The reason we want cover on orchards is because it's really, really important that roots of trees stay cool. Fantastically important. Same with vines. When they stay cool, and this is true for all plants, by the way, when we have the soil well insulated, roots stay cool. When roots stay cool, plants can maintain transpiration. When they maintain transpiration in fruit trees, it means that we maintain boron and calcium uptake, which means that for apples, we don't get bitter pit. So it's really important that and also you don't have fruit loss, you have good, your blossom, you don't have blossom rot, things like that. You can actually maintain that, so you want to maintain that stream. Same thing now when we have these really hot temperatures. We need to have that insulation. We need to keep our soil biology growing, and at the same time, that insulation keeps the water in there, but it maintains the soil temperature, and then the plants can cool themselves. And we need them to cool themselves. And we need to take full advantage of the water. So that's just, sorry, I thought about that and needed to add that in. Yes?
You had a slide that had a big green organism. It sounded to me like you said calendula, but that's not what we called it.
No, I called it a collembola.
Calendula.
Correct, collembola.
We got it. Second.
You can call it a springtail.
Okay.
That's easier.
Okay, great. That was interesting to me. Also, if you are making your own soil mix for vegetables, could you give a recipe?
Oh, making a soil.
Components, you can
percentage-wise, okay, if you're going to use, you want to use some soil, but if you're going to amend it, like you're going to amend it with compost, your compost should actually be composted, by the way. Well, people laugh at that, but there's an art and a science to compost making. It's not that straightforward to make good compost. So good compost, don't have it too hot. You want it to have finished. I add anywhere between 50 and 30% compost, like into a raised bed. With manure, I think you have to be more careful. It should be composted manure, because again, everything can get really too hot. And you want to add some of that in the fall. You lose some of the nutrition, but you allow it to decompose if it's not fully decomposed. Because you need to break down, because otherwise your plants won't grow because everything else is growing, and it just, I mean, you'll see that they don't do well. If you're making an actual like soil for like, I don't know, like not raised beds, but even like blueberries or strawberries or things like that, I like to add sand in. I like to have the roots a little bit loose, so I will mix in about 30% sand into my potting mix just to loosen it up. And I don't make it too organic. I like the sandy loam, but I like have compost in there. So, you know, if I'm going to add sand, it'll be 30% sand, probably 30% compost, and then 40% soil. And then I'm going to use, and then I would be using compost tea or a compost extract. And for me, they're almost the same from my perspective. I've got some kind of biology in there that I'm watering with maybe once a week for vegetable beds. And I might be spraying it too, depending on what's happening out there. Like misting, yeah, or misting just for pest control. Yeah. You're welcome. Just a minute, I'll get somebody new, and then I'll come back to you, sir. Yes?
Jumping up on the question also, so and clay soils. So, would you amend what you just said at all?
Are you gardening?
I don't have to amend mine, because I'm always doing something.
Okay. The reason I ask is if you're broad scale agriculture, yeah, it's like, okay, heavy clay is just, you know, I can grow things in heavy clay and grow more roots and then try and break it up that way. My soils are actually really sandy.
And your soils are predominantly where?
South central Kansas.
Oh, okay.
So mine are really sandy. But if it's clay, and I work with a lot of clays in Finland and the Netherlands, and in those clay soils, we definitely grow covers, and we overseed, heavy seed, and we put a lot of roots down, and we pay attention to the depths of root. So we create root canopies, and we are trying to get roots as deep as we can and leave them in there and grow them fairly mature so that we can add organic matter to our clay soils. But organic matter is your friend in clay soils. I mean, it just is.
And I noticed you mentioned Oregon and Montana and a few other places. What are you doing in those places?
For me, mostly I'm consulting on, like, some of it's vegetable, some of it's orchards, some of it's dairies. But mostly I'm helping people grow plant communities that support their agricultural system. Oh, very different. I mean, in Washington State, most of them are basalt. In the northern part of the state, they're basalt soils. So they are very highly iron. But some in the Palouse country, I mean, the soils are beautiful. They're just so lucky to have such good soil, deep soil. On the Oregon coast, volcanic. And as we get down into California, we get a little bit more, we also have volcanic soils and some other rocky soils and whatnot. And sometimes you'd be sitting on a pit that, you know, where you can, in parts of Oregon, you're sitting there and you're sitting on the edge of it and your feet are on solid rock. And there's 1.5 feet of soil. And that's it. There's definitely a hard pan. Okay, just a minute. I've got this man here, and then I'll come back. Yeah.
Can you go into a little bit with like fertilizing regimens and the soil health? Because I've seen a lot about how old fertilization actually it harms the microbial interaction.
It does. Okay, so I like to think about augmenting, and that's why I like tissue testing. So use tissue testing and do it at regular intervals, like get to know when are your key times for the crop that you're growing, okay? And this is about observation. You don't have to measure if you don't want to. Just make sure that you're looking for signs. There actually is, I have it up on my website. There's a really nice resource guide to this that was made by ATP Nutrition out of Winnipeg, Canada. And it has a really nice pictorial, color scheme showing the various nutrient deficiencies that I think is really helpful for people. And you can find those things on apps and things like that. But this one is just happens to be really good and talk about the different, deficiencies and what to look for. Now, the other thing to bear in mind is that if you over-fertilize, it looks the same as deficiency. In fact, they almost look exactly the same. So you have to be really careful about, if you've done a lot of fertility. Nitrogen, plants they need nitrogen, but they don't need over amounts of nitrogen. And so I fertilize a lot with ammonium sulfate. Plants need a little bit of chloride, but they don't need a lot of chloride. And usually we don't use enough sulfur. And I like to use elemental sulfur annually. So I'll put 5 to 15 pounds of elemental sulfur in furrow with my crops. Now, there's a reason I do that, and that's because I want sulfate oxidizing organisms. But that means it takes a really long time to build that population to break down that sulfur. So if I give it a little bit of elemental sulfur every year, then I build up a population, and then by about the second or third year I'm using it, it disappears like that. And it's not a flash in the pan like thiazole. Like thiazole goes in, gets used, and then goes out. When you're growing plants, sulfur is the most important thing they can have. Sulfur and trace elements is the most important thing your plant can have early on in its growing, because that sets its immune system. And so we're trying to have a really strong immune system going forward. We've got good habitat. Let's say we all have good habitat, soils look great. Then we have to make sure we have trace elements and sulfur so the plants grow really fast, they come out of the ground really fast, and they have enough sulfur and trace elements and iron to really have a good, strong immune system. Not a lot of it, but once again, it's just a little bit. And sometimes that foliar feeding and the small side dressing and things like that, instead of putting everything on at once, is a better deal. But I do like to augment. So I'm like one of these people that goes like, I'm going to look at the tissue test, I'm going to augment. If I do my cover crops right, then usually I don't need very much. And I will use just starter fertilizer and everything will grow fine. But, and I don't like to use too much phosphorus because that inhibits mycorrhiza. So I will maintain, I'll be under 10. Sometimes, a lot of times I don't even use phosphorus if my soils have enough phosphorus in them. Like if the total, that's a Bray 2 analysis. If my Bray 2 analysis is like 1,000 ppm of phosphorus, which even then isn't the total, sulfur will get at that. And I'll make that available. So I'll add a little bit in the starter and that would be it. So it just really depends. But yeah, and the other thing is you use too much nitrate, then you actually, you get protein, but the quality of the protein isn't as good. And you lose vitamin C and you lose B vitamins. If you use ammonium sulfate, you build B vitamins. And so, we're looking for nutrition out of our food too, so.
The key is that with this type of regimen, you're doing less regardless.
Yeah. Less is more. Yeah. Mm-hmm. Yes.
So you just mentioned your involvement with the biometer, and I was just wondering, and you said that you were out of Massachusetts area. I was just wondering if you had any involvement with the biometer with Dan.
Oh, dang, I know Dan very well. Yeah. Dan was the impetus for me moving to Massachusetts to co-found Audacious. Yeah. Was I honestly, I don't know. Yeah, but there are other meters that you can use. Yeah, so I'm done, right? One more question?
Is there something, I shouldn't say simple, but it's between the nematode and graphic that's been very distinct from the allelopathic. Is there some place like a resource that it might be an oversimplification, maybe it is.
Yeah, there is. SARE still has the managing your cover crops profitably. And there are charts in the middle of that that tell you a little bit about what some of these plants do. Yeah, and I promised people that I would work on putting some of this stuff together, and I am working on it, and eventually we'll have it up, and I'll run a course on it at some point in time. But yeah, these are multi, as you can imagine, you start getting into this stuff, it's a multi-day workshop, so yeah. One more then, that's it, I have to cut off.
You talked about the seed and the sprout and that seed being strong for 10 days. So many producers are using NPK and S for starter fertilizer. Other guys are starting to add magnesium and calcium and different things. What's your thought on adding more nutrients to that starter as opposed to just those four?
I would always add more. I put my traces in there too. I mean, but again, trace. This is not like, and don't think that it's going to last the whole year. It's not. Just make sure the plant gets a good start. But you have to be careful. So there's one caveat I'm going to say with that. You add too much and the plant has to give up water and it has to give up some of its ionic balance in order to balance the ions that you put in there. So it's a really fine, when a seed's germinating, be careful about the salts and things like that, because if it's salty, then it will have to give up water, it will have to give up other things, and you'll actually hurt the germination and establishment of that plant. So it's a really, you want to just do spoon feed it, here, have a little bit of stuff, just to make sure. Okay.